Remember, too, that I do have an extra credit opportunity I told you guys about the other day. It's the Toys for Tots. My office is in the building, obviously, where we have lecture and lab. It's room 123. So if you come in those main doors, you come in that lobby area, you come in those big doors, immediately to your left, there's two wooden double doors. And if you open that, you see my office. I have a big bear standing in the window. A stuffed black bear. Good morning, Aura. How are you? It's kind of chilly this morning. So if you do drop off a toy, by the way, make sure that you put your name on my door. There's a list. Make sure you put your name and what you donated. Okay. And you can donate as many toys as you want, but I'm only going to give you 10 points for you don't get 10 points per toy. Okay, so now in chapter 15, we're going to start really drilling down into what's going on with the chromosomes, and we've talked about the results of it, with Mendel's work. We've talked about how if you cross two heterozygotes together, you get the phenotypic ratios and all that sort of stuff that we talked about in chapter 14. Next week in lab, by the way, we're going to do the last lab in your lab manual, the one you downloaded. I think it's lab 11. It's the one that has all the genetics problems. Also, make sure you practice or have done those practice genetics problems too. So if you have any questions, we can entertain those questions in lab too. That's kind of basically what we're going to do in lab. We're not going to be doing anything really as much as we're just going to keep practicing doing genetics problems, different types of them. So, familiarize yourself, try to go through those. And if you have any questions or confusions, that'd be a great time to bring it up. But in chapter 15, what we do is we kind of turn our attention to what's going on, in terms of the chromosomes and how that affects, like I explained to you briefly with the flowers, with Mendel's flowers. You remember, if you were a purple flower, what it is the genetic information that you had, that those A's and T's and all that stuff that we talked about from way back. All of that genetic information there contains information about a trait, and the color of a flower would be a trait, just like the color of the seeds was a trait, and then the round and wrinkled and all that stuff we've been talking about, right? But remember I told you that with the purple flower, that because of the genetic information, an enzyme was made that synthesizes that purple pigment, that color. And if you're a white flower, then your genetic code is a little bit different, and you don't make that enzyme. Right. So what we're doing now as we get through these last few chapters is we're starting to kind of go full circle. Honestly, we're going back to how those proteins were put together. You remember way back in chapter, oh God, I don't remember, the organic molecule chapter, when we talked about the carbs and the lipids and all that stuff. When we talked about the proteins, we spent an awful lot of time talking about their shape, didn't we? And we emphasized that again in enzymes. We talked about it more than once, that the shape of a protein is so vitally important. And at the time, if you remember, we did the 4 levels of protein. You remember the primary and the secondary, the tertiary, and the quaternary. Now, if you remember those 4 levels, and I'm hoping you remember at least some of it, does anybody remember what the primary level of protein structure is? If someone asked you to define, what is the primary level of protein structure? What does that mean when someone says that? What would you tell them? And hello to those who are signing in. Good morning. Yeah, the main structure of all proteins, the primary level of structure. Yeah, it's the amino acids. You're right. And not just the amino acids though, but remember it was the order of the amino acids, right? It was having the right amino acids in the right order. Make sure that you get the right shaped protein, because you remember the thing about amino acids, they all got stuff hanging off of them. They got all these side groups, and some of those side groups attract each other, and some repel. And then some don't like water, and the world's a really water place. So no matter where you are, in the ocean, in a cell, you're surrounded by water. You remember when we talked about osmosis a few chapters ago, and we said that the average solute concentration in a cell was 1%, right? The average solute. That means 99% of a cell is water. Okay, so water is everywhere. So water attracts certain side groups of those amino acids and repels others, and whatever the case is, it folds it up. Remember, primary, secondary, tertiary, all that's about folding. And even though that shape that ends up looks like a blob, it's a very particular blob to Mother Nature. Here's what we're doing with the next few chapters. We're going to look down at exactly how that happens, exactly how that right order of the amino acids is put together. Remember, even at the time I said the information on how to do that, the recipe on how to make a protein, the primary level of sequence, that's contained in that last organic molecule, the nucleic acids, the DNA and the RNA. So that's kind of where we're going with that in sort of an overview. And that's going to kind of guide us actually pretty much through the rest of the semester. You know, we've seen the results of with Mendel's work. So now we're going to start really drilling down. Now, this is a cool picture to start with, too. This is a karyotype. Remember, a karyotype, a karyotype was a picture of chromosomes. And I think I told you at the time, remember when I was in college, we weren't all fancy and stuff. We had photographs of karyotypes. And when you take a picture of the chromosomes in a cell, of course, they're condensed, so they're easier to see. But you know, they're not necessarily arranged next to the homologous. So what happens with that is, like, you cut out these picture of all these chromosomes in prophase all thickened up like this, and then you try to match them. Remember I told you that was a pain in the butt to do. Now they do it with computers. Your book says this is the number 6 human chromosome, and specifically what you're looking at in yellow, and this is what's so cool about making stuff fluoresce. They actually use the chemical reaction to make certain areas, and what's fluorescing there are genes. Okay, that's what gives you the recipe for a protein, folks. That's where the connection is made, the gene. Years ago, they used to always talk about the one gene, one enzyme hypothesis in genetics. And basically what that says is enzymes and proteins in general, right? All proteins have that primary level of amino acids, and that recipe is the genetic information. That A's and T's and G's and C's that we learned about a long time ago. That sequence of letters is going to mean whether you make that purple pigment or not, whether you're tall or short, what color your hair is, you know, everything about you, that sequence of letters. That's what's so cool about this, right? It's like a decoder ring is basically what you're looking at. Now, before we go on, though, I want to see how much you guys remember from the chapters where we talked about cell division. You notice that you got 2 yellow dots there, right? And I just told you what a yellow dot was. It was a gene. And of course, now make the connection. A gene has alleles, remember? Right? Genes control for a genetic trait, and alleles are alternatives. So purple and white would be alternatives for the trait that's controlled by the flower color gene or the seed color gene. So looking at that picture right there, why do you think there are 2 yellow dots? Why do you think there are 2 genes? What does that tell you about the state of these chromosomes? Or maybe they're not just chromosomes. So you guys put on your thinking caps early in the morning for that. Actually slept in today, almost 6 o'clock. Crazy. So think about it. You guys typing, thinking, or are we just having a slow computer time? Good morning, Clarissa. How are you? Oh, that's right. Or you get up early. That's true. The question that I'm asking is, if you're looking at this picture, this is obviously chromosomes, right? These are prophase chromosomes. They've condensed. And the yellow spots are genes. I want to know why there are 2 yellow spots. Now the yellow spots, you know, since they're across from each other, if you look at it, they're the same genes. No. Although that is something we're going to talk about, yeah, X and Y. No, that's not what we're seeing here. Remember the term. Remember the definition of an allele, an alternative form of a gene. Now, sometimes those alleles are the same in your homozygous. Sometimes they're different in your heterozygous. But no matter if you're homozygous or heterozygous, how many alleles are you going to have if you make a copy of a chromosome? Can you see how many? Can you see how many different? See how many molecules there are? The number 6 right there. Can you see how many molecules there are? Can you see that there's more than one molecule here? Does that help? That's more than one. That's more than one nucleic acid. There's one here, and then this one is a copy. So you think, what was the question again? The question was, why are there 2 yellow dots? What does that tell you about the state of these chromosomes? Or maybe if you're, to kind of think about just terminology, these are chromosomes, of course, but what else are they? Because of the way they look right there in that picture. They're not just chromosomes, they're also, sounds like, there you go. Thank you. Yes. Yeah, that's right. They make copies of themselves, right? Do that little detective work. That's when you look at that picture in your book. Don't just scan over those pictures, because sometimes they have some really interesting information. And what that does, folks, is it helps round out your knowledge. You look at that, and the book is telling you why it's got 2 yellow dots there, right? So you look at that and you go, oh, right. Because that's what happens when chromosomes make copies of themselves. They make copies. And so these are 2 sister chromatids. Isn't that kind of cool when you do that? It's like that, wow, that's so cool. It's what I call an ooh-ah fact. You know what an ooh-ah fact is? I tell you something, you go, ooh, wow, that's so neat, right? Hopefully you've done that a couple of times this semester. All right, yeah, R mentioned X and Y chromosomes. So let's talk about that for a second. Let's talk about what is collectively called the chromosomal basis of sex. So how sex is inherited. And you might be surprised, or maybe you won't, or maybe you'll be ooh-ah over it, honestly, is there's more than one way for animals to do this. And I'll show you in the next slide a couple of different ways. But us animals, human animals, we kind of do the typical route. The typical way it's done in human beings, and I'm sure you're very familiar with this, to be honest, is you have homologous sex chromosomes, just like you have every other pair of chromosomes, just like number 2 paternal has a number 2 maternal, right? All of those things that we talked about in meiosis, the sex chromosomes that you have, and you only have one pair of sex chromosomes. So basically what we say among organisms is they have one pair of sex chromosomes. I'm typing this out, okay? And then they have all the other ones, which are called autosomes. So in human beings, we have one pair of sex chromosomes and 22 pairs of autosomes, right? 22 pairs of autosomes plus a pair of sex chromosomes, and that makes us up 23 pairs. So you understand the difference between autosomes and sex chromosomes, right? Autosomes are like the number 8, the number 5, all that other stuff. The sex chromosomes also aren't going to be numbered. We don't have a number 6 paternal and a number 6 maternal. We have an X and a Y. Now, they are related to who you got them from, which we'll see in just a second. But that's how it works in human beings, lots of other critters. If you're an X and a Y, then you're a male. And if you're an X, whoops, too many X's. If you're an X and an X, you're a female. And this is a really cool picture. Again, these are obviously chromosomes that are undergoing cell division, because you can see there's sister chromatids there, right? Well, there's the X chromosome, and the Y is basically it was actually a book written years ago called The Fragmented X. And basically what it says is perhaps what happened through evolution. This says something kind of interesting about, the chromosomal basis and the fact that sex even exists today. That maybe what happened during evolution is an X chromosome split up and left just a little piece behind. That's what the Y is now. And one of the bits of evidence for that is you would think, okay, well, during synapses, when the homologous chromosomes seek each other out, the way they know they're homologous, of course, chromosomes can't speak, is they have parts. They have parts that, like a magnet, would stick them together. So by virtue of their genetic code, they have areas in which they'd actually be attracted to one another. The Y chromosome, even though only being half of the X, let's say, or fragmented, it has those, that's what it says, they're homologous segments. It has those chunks, those little magnetic chemical parts of its DNA. It's not really, well, it is kind of magnetic, electrostatic, okay, that attract the X and the Y together. So that's why they synapse during meiosis, even though they're not exactly, it's like homologous, are they? One's a lot shorter than the other, even though they really are homologous. Now, the reason why having a Y chromosome makes you a male is because we found that there's a gene on the Y chromosome called the SRY gene. When you discover a gene, you can give it a name. You can name it like if you discovered a species. Sometimes genes have like a name, somebody's name attached to them. So maybe somebody they're trying to honor who did a lot of work in genetic research. And sometimes what they do. This is SRY is the sex determining region gene. See, the sex determining region. The Y part is not part of the word, but that's the Y chromosome. And what the SRY gene does is it controls testes development. Okay, so if you have the X chromosome, like if you have two X chromosomes, then you don't have that SRY gene, so you don't develop testes, obviously, so you're a female. Most of the time. I've got some interesting chromosomal things that I'll talk about later on about what happens when chromosomes don't do exactly what they're supposed to do. Here's what I was talking about, the fact that there's more than one way to do it, literally. Human beings, we do it, as you see up the top. The individual on the left has got 44 autosomes or 22 pairs. Okay, I know they switch back and forth, and that, but you understand. And then a pair of sex chromosomes, and since you're XY, you're male, and you're XX, you're female. So now during meiosis, okay, so what we're seeing in that first arrow right there is pick a different color than white, is meiosis. All right, so during meiosis, we're going to have the number of chromosomes, so we get now 22 or 11 pairs, all right? And you either get an X or a Y if you're a male. And it's kind of 50-50. I think it's one of those things where, if I remember this correctly, in most cases, males during ejaculation produce more Y sperm than X, but I believe X sperm or Y sperm have a greater mortality. In other words, more Y sperm die off before they get to fertilize the egg. So the sex ratio of boys and girls being born is, in humans, I think it's pretty close to 50-50, okay, because of that. All right, so then what we're looking at down below, and of course, and the female on the right, she can only produce eggs with one type of sex chromosome, right? She can only, since she's an XX, no matter which egg you get, you're going to get an X chromosome. So it's the interesting thing, right? And I wrote it to the right there that in us, it's the sperm that determines the sex, right? Because down below are the zygotes, or the offspring. So if it's an X sperm that fertilizes an X egg, you get an XX, and that's a girl. And then, if it's a Y, then you get a female, or you get a male. Isn't that cool? I think that's kind of neat, especially when you think of history. Remember the old English kings, or maybe other royalty, did that. Who was responsible for giving the king a male heir? Got to have a male heir. Couldn't have a female. God forbid, right? Had to have a king. But who was responsible for doing that? And I say responsible in quotes, because you can't be responsible for this. But who was supposed to give the king male heirs? Yeah, that's right. All along biologically, I understand they didn't understand biology at that level back then. Didn't quite give you the excuse to do that, but nonetheless, it's ironic. Now, as I said, there are other critters that do it a little bit differently, like insects down below do something very interesting. It's still the sperm that decides whether you're male or female, but in this particular case, it's not what the sperm necessarily, brings you. It's not a Y or an X chromosome. In the case of many insects, it's called the XO system. There's no O chromosome, but what we mean by that is if you're a female, then the sperm contained an X chromosome. But if you're a male, the sperm contained the autosomes, but no sex chromosome. All right, so it's not what sex chromosome, either yes, you got one, or no, you didn't. You got all the other autosomes. It's a little bit different than like in the bees. We'll jump to the bees. Because in the bees, it's really kind of cool. If you're female, you're diploid. If you're male, you're haploid. So males only have half the number of chromosomes. So they kind of took that exosystem and went And there are reasons why that is. If you know that bees have a very social society, and most males are worker bees. Some species of female bees can actually fertilize their own eggs. Some species of lizards can do that too. Some species of organisms have no males. Do you know that? Some species of organisms that have what we technically call sexual reproduction, there are no males. It's only females. Lizards, fish. Oh, tell you a cool story. Up at the Shedd Aquarium in Chicago, if you've never been to the Shedd Aquarium, if you're going to Chicago, God, go see the Shedd Aquarium. It's incredible. But they had a sand tiger shark in their collection. They had one female sand tiger shark, and she had been there a long time, a decade or so, or something like that. Been quite a while. And sand tiger sharks, if you've ever been to SeaWorld, sand tiger sharks are very popular in aquariums because they look really ferocious. They have really snaggled shark's teeth, but they really are kind of tame, to be honest, relatively speaking. Not that I'd want to get a tangle with one. They're not known as man-eaters. Anyway, so this sand tiger shark, this female who's been in this aquarium for 10 years, one day the aquarists walk in the morning and they find sand tiger shark pups. She's given birth. They're like, there are no male sand tiger sharks in the exhibit, and there are other species that are male, but species don't generally mate with one another, right? And these didn't look like they were hybrids of anything. They looked like young sand tigers. Little did we know at the time that even though that we knew certain critters, these little microscopic critters called rotifers and lizards, we knew that some of them could do that. We weren't aware that something that is big, if you will, or as advanced, if that's a good word, as a shark could do that, right? That was kind of interesting to us. That female sand tiger shark had actually been able to fertilize her own eggs. Even though there was no male around, she created eggs and fertilized them. Now, if you're thinking, oh my goodness, all the inbreeding stuff, yeah, that happens. But in the absence of a male, inbreeding isn't 100%. It sounds awful, but if you have an incestuous sexual relationship with a close family member, it doesn't guarantee the child will be born with deficits, okay? It heightens the probability pretty good, but it doesn't guarantee it. So if you're those organisms that do that, the way you get around the problem of the inbreeding, what we call inbreeding depression, that would be a problem offspring. So you have lots of them, right? So if you know that 20 out of 100 are going to have a problem, have 100. You know that 80 of them will survive. So sometimes some animals, they play a lot with numbers here. Now, among the chickens, it's the female that determines the sex. Isn't that kind of cool? So in the case of a female, or in the case of the chickens, if you inherit a Y chromosome, you're a female. And if you inherit a Z chromosome, then you're a male. Now the letters, I don't know, maybe it looks like a Z or a W. I'm not sure. The X and the Y kind of, well, looks kind of a Y with the tail missing, I guess. I'm not sure where the letters come from. Isn't that interesting? And there are variants of this, by the way, but there is easily said more than one way to do it, literally. All right, now the next slide I want to talk about. When we think about sex chromosomes, we think correctly. They determine sex. They determine what we call primary and secondary sexual characteristics, and you actually might be surprised to find out, you know, that maybe what you think are your primary sexual characteristics really aren't. Your primary sexual characteristics are whether you have ovaries or testes. Everything else, your genitalia, etc., are secondary. Okay, that's the delivery device or the receptive device. It's the ovaries and testes that are important for all of this. So the sex chromosomes contain a lot of information about sex. Yes, they do. There's no doubt about that. But for some reason, and considering a lot of our chromosomal information is, we think, just junk, or a lot of it's repetitive, a lot of it does functions that really don't have anything to do with proteins. A lot of it does no functions. Mother Nature has chosen somehow through evolution to put a few genes or traits onto the X chromosome that have nothing really to do with sex. Okay? In other words, the genetic trait on the X chromosome really doesn't have anything to do with making you male or female. It's like Mother Nature found and just saying, okay, we'll take that genetic trait and just put it over there. All right? She didn't put it on an autosome. She put it on a sex chromosome. Now, what this leads to, is something we call X-linked characteristics. right? So in other words, what we're saying is characteristics that human beings and other organisms can exhibit, that you exhibit, phenotypes, okay, things you have, that have really nothing to do with what sex you are, but show up in a higher proportion in a sex versus one. All right. So, for example, I'm going to do the slide in just a second, but it contains a lot of information on it, and I kind of wanted to lead you a little bit further along before I hit you with this slide. Does anybody know anybody who's colorblind, or is anybody colorblind in class? We actually have a test that we'll do next week that we use, a very simple test to detect colorblindness. But you know someone who's colorblind? And I'm not talking about what my wife calls colorblind, because I don't know the difference between taupe and brown. No, the taupe person. I'm like, what color is taupe? Or no, the fuchsia top. I'm like, the purple one? Why can't we just have, you know, half dozen or so colors? So I'm talking about the typical red-green color blindness, which is there's other kinds, but that's the typical one. Nobody knows anybody who's colorblind? Nobody in the family? Nobody they've ever met? Does anybody know which sex is more likely to be colorblind among humans, males or females? Yeah, Jessica, male or female? Okay. Anybody know someone? Yeah, and that could be, like I said, there's more than one kind. There's some rare types. The most common type is what we call red-green. Does anybody know a female colorblind person? Red-green, red-green, because I'm not as familiar with the inheritance of the other types. No, nobody knows any females. Now, it is possible. Okay, well, the observation that was made by geneticists was that a trait colorblindness, which, come on, I mean, you know, males are always accused of being colorblind, but that's the taupe brown thing I was talking about, I think, more than anything. But, you know, the red-green colorblindness, it was obvious that it was more common in males and females, and we couldn't quite figure out why. Well, the reason being is because colorblindness, and there's a couple of other disorders I'll show you on the next slide, are actually, the trait for whether you have it or not, is found on the X chromosome. So that's what we call X-linked, but then it's like, it really doesn't have anything to do with sex. Being red-green colorblind doesn't have anything to do with your primary or secondary sexual characteristics. So why is it that way? So let's take a look at this this slide now. Now, some other traits are X-linked too, by the way. A type of muscular dystrophy known as Duchenne, and then hemophilia, the folks whose blood doesn't clot. They can actually bleed to death by simply bumping into something. All right, so what we're looking at above, and let me kind of familiarize you with the terminology. So we're talking about an X-linked characteristic, so we're just going to be looking at the X chromosomes, so we're not looking at the autosomes. And if it's a capital N, then it's dominant, obviously, and if it's a lowercase n, it's recessive. Now, one thing I got to say about dominance and recessive before we go on. Most recessive traits that are detrimental, I'm sorry, skip the recessive. Most traits that are detrimental, red, green, colorblind would be detrimental. It's probably especially during the caveman days, okay? But most of those kind of traits that would be detrimental, hemophilia, that's definitely detrimental, muscular dystrophy would be detrimental, are recessive disorders. And what I mean by that is you're not going to have that unless you're homozygous recessive. All right, so the only way that a female could be colorblind, female, right, is if she has two recessive alleles. All right, makes sense. So the odds of that happening are going to be a lot less, because look at the male now. Now look at this box that's colored red. This is what we call a pedigree, and the red, the dark color, sometimes it's not red, I don't mean to focus on just red. In this particular way they're doing it, the darker the color is going to be referring to organisms that have the trait, and the lighter color is going to be organisms that are carrier. Now there's a very precise definition for carrier, folks. In genetics, a carrier is an organism that is heterozygous, that carries the trait, carries the allele, but does not exhibit the trait. You see the females in the box in the Punnett square down below that are this way? Are they colorblind based on what I just told you? That's right. They're not colorblind. They're not colorblind, but they carry it. Okay, now jump back up to the male. Notice it's the darker color. Why is he colorblind then? He's only got one X, or doggone it. He's only got one N. Scratch that part I just said. Why do I do that? I did that the other day in Bio 2. Brain gets ahead of my mouth. Why does that poor male up there who's an XY, why with only one N, with only one recessive allele, why is he colorblind then? The females only have one recessive allele. They're not colorblind. They're carriers, but they're not colorblind. I know I blurted it out. That's fine, but type it in there. Why do you... This is an important concept to understand about X-linked characteristics. When you're working your genetics problems, a lot of the genetics problems are X-linked genetics problems. So again, why is that male colorblind? Yeah, that's true, but concentrate more on the individual itself. Concentrate more on that guy in the block. Why is he colorblind? Why is he colorblind? And the females that only have one recessive alleles, these down here, why aren't they? The answer, you're probably making the answer too difficult, folks. Step back for just a second and look what's different between the male and the female as it comes to their X chromosomes. You remember the definition of a recessive allele? A recessive allele only expresses itself when it's by itself. Yeah, there's no other allele. Yes, there's another chromosome, but it's a Y. It's a different chromosome. Remember, yes, they do go into synapses. Yes, 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 all that, but it is not the same chromosome. It doesn't have maybe that part where the genetic trait of colorblindness would be found. So if you're a male, the reason why these traits, colorblindness and hemophilia, they're all the same as far as how they're inherited. The reason why it's more prominent in males is males, you got one shot. You get one bad X. You get one X with a recessive allele. Just one. You don't have another one, right? As Marie pointed out, you don't have another one for it to be dominated by, okay? So that male up there has got that one recessive allele, that little N, and he's going to have the trait. Now, if you're a female, you can have one, and you're cool because of the fact that you won't have the trait, but you are going to be a carrier. Okay? So here's what this says. You got the colorblind male and the female on the left that he's mating with. So I'm starting up at the top. She has normal vision, completely normal vision. She's not a carrier. A colorblind male is going to produce no colorblind sons. That's an interesting fact, right? Look at the Punnett square down below. We just learned that it's the male who determines the sex. So if it's a son, it's because he gave the Y chromosome. No problem there. That's not where the trait is. Half of his daughters, though, 50%, or there's a 50% probability that they'll be carriers. All right, now they mate down below, like you see on the left-hand side. All right, take those carriers and change the fathers. So in one case, we have a father with normal vision. So mom's got normal vision, right? She's a carrier, I know. Dad's got normal vision. Half of their kids will have normal vision, male or female. There's a 25% chance that the child is going to be a carrier for it and a female. And there's a 25% chance that you can have a child who's a male who has the trait. Or if you just want to separate it amongst the boys, half of your boys are going to be colorblind. Now, you notice that the Punnett square down below is set up just like we did before. We're just separating the alleles on sex chromosomes, right? So this is the columns and the rows just like we did before. If, on the other hand, a carrier female and a colorblind male mate, then you got half of your children are going to be colorblind, and a quarter of them will be carriers, and only a quarter will have normal vision if they're male. All right, this is important when trying to calculate pedigrees. For example, I always say color. Let me erase all my scribble. colorblind daughters have to have colorblind fathers. It's an easy way to analyze pedigree. Colorblind daughters, think about what I'm saying, colorblind daughters. All right, if you have a pen or you have a touch screen, write the genotype for a colorblind daughter. In other words, what are her X's going to look like if she's colorblind? Or you can type it in the chat box. Don't worry about the superscript part. I'm just looking for the right letters. So we got a colorblind female. Okay, we got a colorblind female. And now go back and tell me how that colorblind female ended up. What do we know about her father? Color. There we go. Correct. Okay, right there. Colorblind females have to have colorblind fathers. Why? Two things are going together there. It's like a synergy thing. Number one, if you are a male and your offspring is female, you contributed the X chromosome. If you'd have contributed the Y, she wouldn't be a she, she'd be he. It'd be a son. But you contributed the X chromosome. So if that daughter is colorblind, that means that the only X you gave her was colorblind. Therefore, you must be, because you only got one. And mom's got to be colorblind or be a carrier. Now, mom and dad are both colorblind. The whole doggone family's colorblind. Everybody's colorblind. Do they test for colorblindness in school anymore? They used to test us. Did anybody get tested? They would have held, and we're going to do it in lab actually, held up these pictures of different colors, and there's supposed to be numbers in amongst the pictures, and you're supposed to see the numbers or not. I don't know if they test for that anymore. They did then also when I went in the military, they tested me for that. Nobody ever was tested for that in school? I guess they probably wait to see if it's obvious. It would become obvious when you start driving, that's for sure. Unless you remember the light. I guess you just remember the red is above. Okay, anybody ever have a calico cat? Calico cats. Calico? Not a tortoise hair color, but a calico cat. Calico cats are typically females. There are cases when they're males, but if they're males, there's actually something wrong with them, as I'll hopefully remember to tell you as we get toward the end of this information. Here's how a calico cat forms. A calico cat forms because we get a mosaic of genetic information, all right? Calico cats are typically female, so they have two X chromosomes, so we're starting at the top. So what happens is on one of those X chromosomes is the allele for orange fur, and one is black. So this is a heterozygous female. So those are conditions that have to be met. In female mammals, what happens is something called X inactivation. In other words, one of the X chromosomes in you as well becomes inactive. It shrinks down and it becomes inactive. This is what we call to balance dosage. In other words, it's like Goldilocks, right? You don't want to make too little of something if you're a gene. You don't want to make too much of something. So in the case of females, having one X chromosome is enough. You want to inactivate the other one. Now, the reason why calico cats end up this mixture of orange and black is because it's random in which cell that X becomes inactivated. So in the case of the cat, right, in some of the cells, it's the orange fur allele X that shrinks, the one on the left, and in the orange, it's the other one. All right, so the inactive X doesn't get expressed. This inactive X, by the way, you can see it on under a microscope. It's called a bar body. Same thing. Probably after the first person who described them or discovered them. Now, what effect does this have on humans? Well, like I said, it can have effects based on dosage limits and dosage requirements. But it happens normally in everybody. All you females out there have the same thing. One of your X's has become inactivated. Now, when they go through meiosis, they reactivate. So, in other words, they spring out again, replicate, go through the process, and then again, randomly, they shrink back up. Only need one. Cool experiment here. I like that they put this in the book. Question oftentimes is, why is blonde hair and blue eyes seem to come together? Folks with blonde hair very often have blue eyes, and there are other traits that fit that category. Well, it has to do with something called gene linkage, and it actually has to do or caused by crossing over. Way back in prophase 1, you remember when I said the genes cross over? Now, the closer the genes are together on the chromosome, typically when homologous chromosomes come together, they only cross over 1 to 3 places. Now, it's different each time and all that stuff I talked about. But if the genes are closer together on the chromosome, when they cross over, those genes are often inherited as a group. So with the blonde hair and blue eyes, and actually blonde hair is controlled by many different genes, but if you inherit the blonde hair genes, since the blue gene is very close, it's physical proximity on the chromosome to it, you often inherit those together. Now, the reason why we know this is because we've done some research on it. We've collected some data. Take a look at this cross that we're looking at here. These are fruit flies. Fruit flies are a very common genetic critter that use. And when they say wild type is going to be the dominant. All right, wild type is going to be the dominant phenotype. So the dominant phenotype in fruit flies is you're going to have a gray body and normal wings, and the recessive is going to be you're going to have a black body and shorter vestigial wings. Okay, so look at the cross you got up above. You got a homozygous dominant on the left. Now they're not, I know they're not using uppercase and lowercase. Remember I said the symbology can change, but now the plus means dominant. And if you don't have the plus, then you're recessive. So they got a homozygous dominant, doubly homozygous dominant on the left, doubly homozygous or doubly just recessive on the right. Okay? Now you know what that's going to make. That's going to make this offspring right here. Right? You're not going to have any black vestigial wings. You're not going to have any gray vestigial wings. You're not going to have any black normal wings. All of your offspring are going to be heterozygous, and since the plus is the positive, they're all going to be gray, and they're all going to have normal wings, right? No problem there. If you need to understand that, put all this into a Punnett square. Put these alleles into a Punnett square up above, and you'll get the answer that you see right there. All right, now we're going to take that one, and we're going to do a test cross. Remember what a test cross does? A test cross tells us whether, well, in the case of humans, a test cross would tell us whether a brown-eyed person was homozygous or heterozygous. No need to test cross that genotype, folks. Remember, that genotype is the only one whose phenotype, blue eyes in this case, you're going to know. It's these individuals you don't know. So in the case of the fruit flies, we don't know whether a gray fruit fly or one with normal wings is homozygous or heterozygous, so we're going to test that. Remember how we set up a test cross is we say, all right, some brown-eyed individual, we're going to mate it with something whose genetic information and genotype we know, so we're not going to pick another brown-eyed. We're going to pick a blue-eyed. And if all the offspring are brown, then that's probably homozygous. And if half of them are blue, right, that's what the test is all about. Hopefully you've been practicing those genetics problems and know what I mean by a test cross. So that's what they're doing there. Now, if you have been working on those problems, you know that in a test cross, if indeed the organism that you are testing is heterozygous, you'll get equal proportions of all the possible phenotypes. So in other words, a quarter of them will have the gray bodies and have long wings, okay, normal wings. A quarter of them will have the black bodies and have short wings. A quarter of them will have the gray bodies and short wings, and a quarter of them will be messed up, will have the black body and the normal wings. All right, so all four of those proportions, just the same thing we did with the seeds, folks. That that ratio that we got with the seeds of how many were green and round, green and wrinkled, yellow and round, it's the same thing. So this is the predicted ratio that we should get, right? Equal proportions. 1 quarter, 1 quarter, 1 quarter, 1 quarter. So if you have 1,000 flies, 250 of them would be gray and normal, 250 black and vestigial, et cetera, right? Everybody follow me on that? That's the predicted. That's what we would call the hypothesis. Now, it didn't happen that way. You see down below, we got to look at the results. Don't look at the middle part yet. Look at the results. The results are overwhelmingly, we get gray normal-bodied fruit flies, and overwhelmingly, we get black vestigial-bodied fruit flies. We get some of the other types, but we definitely don't get a 1-to-1. We don't get equal ratio at all, do we? We actually get a ratio that's more like everybody is bunched up here, and very few rarities are down there. Now, the reason why that helps us understand linkage groups is we know that we're going to get a ratio down here where they're more lumped up at one end if they're linked together. Because we know that if you typically inherit, like during crossing over, if you end up being the offspring that gets that cell that during crossing over inherited the gray body, you typically are going to also inherit normal shaped wings. So gray and normal, those two traits must be pretty close to each other on the chromosome. Gray and vestigial must be much further apart. Black and normal must be much further apart. Or they're in less of a proportion. So the reason why people get blonde hair and blue eyes, those genes are linked to each other. They're close to one another. We don't mean that they are physically linked. We mean that when you inherit them, they typically come over linked together. Beautiful science there. Very cool. You didn't get the ratio you expected, therefore something else must have went on, and they were able to explain it. And if you understand, and there's more discussion in your book about linkage groups. I don't expect you to know that, but there's more evidence that puts these two together on the chromosomes just in the ratio that we got there. Okay, sometimes it doesn't happen right, and you end up with an abnormal chromosome number. Either you get one off, one too many or one too few, or you'll find out that you can actually inherit a whole nother set of chromosomes. So you won't be diploid, you'll be like triploid, for example. I'll show you that in the next slide. Let's first talk about why that happens. In other words, why do you get the wrong number of chromosomes? Let's go back and remember what we know about meiosis. So let's look up here at meiosis 1. You remember one of the checkpoints in mitosis was the M phase, and that's when the cell makes sure that each chromatid has a spindle fiber attached to it. All right, so it goes through and it makes sure on the chromosomes that each one has a spindle fiber. And obviously, for the reason if you don't, then you're not going to be able to pull them apart during the anaphase. Well, let's say that M phase doesn't detect a problem. In other words, let's say this spindle fiber over here is missing. What you're going to get is a really cool word, actually. It's called a chromosomal non-disjunction. What I think is cool about that word is it's like a double negative. So junction is to come together. Disjunction is to come apart. Non-disjunction is you didn't come apart. So in other words, what we're basically saying, in a non-disjunction, something got screwed up in anaphase. In anaphase, these should have split apart, and they did not. It happens, right? Mistakes are made. So then we'll jump down to meiosis 2. We'll look at the cells on the right in just a minute. So you go into meiosis 2, and the chromosomes split apart. And what you end up with when you finish with meiosis 1 is all of your sex cells, all your gametes are going to be messed up. Half of them will have an extra chromosome, half of them will be missing a chromosome. So having a non-disjunction in meiosis 1 is the most detrimental, right? Because again, it's the Goldilocks. You're supposed to have the right number of chromosomes. If you look on the right-hand side, though, if the non-disjunction occurs in meiosis 2, so if meiosis 1 works correctly, then you got a 50-50 chance of having a good egg or sperm or good gamete, right? See, the ones on the right are normal, they're N, and the ones on the right or on the left, and here, half of them will have one chromosome too many, and half of them have one chromosome too few. So again, that does happen. It's not often, but it does. But I suppose if it was going to happen, you would obviously want it to happen down here, because you're going to at least 50% of your gametes will be normal. Okay, so what happens if you have an excess or a deficiency of a chromosome is called an aneuploidy. See, I put it there mathematically because not everybody's the same number of chromosomes as us. But if this was a human being, an aneuploidy would be someone with 47 or, in this case, 45 chromosomes, right? Normal's 46. And if you get a non-disjunction that goes all the way down the autosomes, in other words, you get no spindle fibers forming on one side at all, which does happen, you can end up with someone with 69 in the case of humans, or 92 chromosomes, a triploid or a tetraploid. Now, oftentimes those fetuses are spontaneously aborted, okay, or miscarried. But if those individuals are born, and they do happen, not just in other animals, but also in humans, they have severe physiological and mental challenges. Those folks aren't going to, unfortunately, lead a normal life by any stretch of the imagination. And what it's caused by, again, is a non-disjunction, either in the sperm or the egg, or in the worst case scenario, it's going to happen in both of them. An aneuploidy, one of the aneuploidies that you might be familiar with in terms of have heard about, is called Down syndrome. Down syndrome affects about 1 out of every 700 children. That little girl in the picture there is exhibiting what we would call the phenotype for Down syndrome. And Down syndrome has another name. It's called trisomy 21. Which literally means 3 number 21 chromosomes, Sam. That's how you get Down syndrome. So some sort of a non-disjunction occurred and caused an aneuploidy. So an egg had an extra X chromosome. Or it seems to be more often in females as they get older. We're not sure exactly the mechanism why, but we notice, and if you look at the chart down below, the incidence of Down syndrome per 1,000 births rises exponentially after about the age of 32 or 33. One of the reasons we believe that may happen is because females produce all the eggs they're going to produce at birth. When you're born, if you're female, you already have all your eggs throughout your entire life made. All they're going to do is, when puberty starts, is every 28 days or so, they're going to ovulate. The older you are, essentially, the older the egg is, the better, the greater the probability it came into contact with some carcinogen or cosmic ray or radioactivity that might have mutated it, and so caused it to have a messed up spindle fibers, a non-disjunction, and then you have an aneuploidy. However, we are also finding out that it also can occur in higher rates among older males. That even though we make sperm on a continual basis, right? So we're always essentially refreshing our sperm. When you're older, there's a greater chance for meiosis to get messed up, to have a non-disjunction. So males aren't totally out of the loop when it comes to higher rates of Down syndrome. Now, trisomies can occur in other chromosomes as well. There's a trisomy 18. I'm not familiar with the characteristics of that. Oftentimes, aneuploidies are going to end up, like in polyploidies, going to end up in a spontaneous abortion, not just in female, or not just in humans, but in other creatures. One place where it seems like aneuploidies are a little less detrimental is among the sex chromosomes. Okay, you can have aneuploidies in the autosomes, and you can also have them in the X and the Ys. Anytime you have a Y, you're going to be a male, though, right? So normal male, extra X chromosome is a Klinefelter's. Extra Y, I guess, doesn't get a fancy name. It's just called a double Y. You remember back what I said to you about calico cats are often, or most often female, although there are exceptions. If you have a male calico cat, you have a very rare cat. The reason why, if you have a male calico cat, you have a Klinefelter's cat. He's male, that's the Y, and since being calico is because one of those bar bodies inactivates, you have 2 X chromosomes. So you can get a male calico cat. It just has to be have a sex chromosome aneuploidy. Double Y was interesting. Years ago, sociobiologists, it's kind of interesting because sometimes it's more science, sometimes it's more, I don't know what to call it, sensationalism. Sociobiology, there was a book out there years ago called The Naked Ape, we being the naked ape, no hair. Basically, sociobiologists are folks who try to explain what we are society-wise by our biology. It's the excuse that cheating males might use on their girlfriends. Well, males, we're supposed to spread our seed, right? Okay. But one of the other things in that book, The Naked Ape, if you ever get a chance to read it, it's interesting. It's by a guy named Desmond Morris. I believe that's who wrote it. But he talks about why women put on lipstick, and he basically says the reason why they do is because it mimics In many species of primates, when the female is sexually receptive, her vulval region becomes engorged with blood. All right, it swells and becomes obvious in color. He claims that's why women paint their lips red. Yeah, I don't know about that. Seems a little bit of a stretch. But one of the things that sociobiologists looked at is if the Y chromosome contains some genetic information to do with aggressiveness, you might expect that higher rates of aggressive males would be double Y. So they went into the prison system and looked at males who were in there for violent crimes, and they found just a slight difference, but nothing that was conclusive. So I guess we say the jury's still out on that. If you're a female, you can have a sex chromosome aneuploidy where you're missing a chromosome. You'd be called a Turner syndrome. But Turner syndrome, Klinefelter's males too, no obvious differences. They might have some breast development, underdeveloped testes, but other than that, they lead a relatively normal life. You may have known or come across somebody who's Klinefelter. Double Y, again, could be some aggressive issues, tend to be taller. Turner syndrome females tend to be shorter, stouter in nature. But again, there are many, many reasons why someone might look that way. And metafemales, both of these actually, one of the things that characterizes them is menstrual irregularities. Now, don't do that third year of med student or med school thing where you start learning out how to diagnose diseases. You know, third year med school, you learn how diseases are caused, and before you know it, you go, oh, I got that. If you're a female with menstrual irregularities, there's a doggone good probability that you're not one of these. Turner syndrome females also, sometimes you'll see online, it says they have a webbed neck. So my inability to draw doesn't help this, but this is, a human. So here's the head, and they're female. They don't have to have long hair. But anyway, what we mean by that is the neck region kind of extends out a little bit further. I'm not saying it goes out to the edge of the shoulders, of course. Again, you probably have more than likely seen somebody who perhaps had this sex chromosome aneuploidies, and you just didn't know it. That's what I said. It seems like it's aneuploidies, like if you have an extra 21 chromosome, you're going to be Down syndrome. There's severe phenotype associated with that. Remember, phenotype doesn't have to be a good thing. It's just a physical expression of the gene. But in the case of sex chromosome aneuploidies, these folks lead a relatively normal life. Now, if you do a Punnett square, you find out where this comes from, right? If we have a situation where a normal male fertilizes with a female that undergoes a non-disjunction, so part of the eggs are XX and part have no sex chromosome, you can get all the mixtures that you see there, right? You can get a Klinefelter. You don't get a double Y. A double Y happens because of a very special type of non-disjunction. A male produces a sperm that has only one Y chromosome, but two Y chromatids. All right, so that's why you get a double Y. Notice down below, though, that there's no such thing as an organism with just a Y chromosome. It can't exist with just a Y, non-viable. In other words, that won't be born. This is in any organisms that are XY. It's not just humans that are XY. Here's a karyotype. If you look down below, I've circled it for you. This is a double Y individual. So this is what I used to have to do, cut them out of glossy photographs and match them all up. And the banding patterns is what I'm talking about matching. If you match them up on length, where the centromere is, notice centromeres in 22, for example, are up here. How darkly patterned they are. Now computers do all this, but you can see that's a double Y. Now, polyploidism, back to being triploid or tetraploid. As I said, among most of the time, especially among animals, that's going to be detrimental. There are some exceptions to this. This is a South American rodent, I believe, that through evidence or through research they found out is closely related to other rodents nearby, but just has double the number of chromosomes. I don't know exactly the numbers. I think your book does go into it. But this is one of the few exceptions where a polyploid, or what appears to be a polyploid, was tolerated. In other words, you do actually have organisms that survive and thrive. That usually in animals, that's detrimental. Usually in animals, that's not going to happen. That's not a good thing. Now in plants, it's totally different. Among plants, it's actually relatively common. If you take my Bio 2 class, you learn that in plants undergoing polyploid, is a relatively common way that new plant species arise. It's really kind of interesting. I guess, I don't know, I guess maybe among plants, this whole dosage balance I was talking about is a bit more tolerated, maybe because they don't have nervous systems or tissues like we do. Maybe they don't have fancy stuff. I don't know. It's kind of like my attitude about buying cars. I rarely try to buy a car with anything fancy on it because they always seem to break. In fact, that power windows. My son says, "Oh, you're so old, Dad. That doesn't happen anymore." But when I was a kid, that was very common. My dad bought a Ford Thunderbird in 1966 that had all this fancy stuff on it and it was always broken. So maybe that's where that comes from. But in plants, like I said, like for example, wheat, what we eat, make into bread. Wheat is actually a grass, a natural, normal grass that scientists have actually encouraged through reasons that I'm not qualified to explain, but through genetic engineering, basically, to engage in polyploidism and then artificially developed wheat. So they made it. So they took a grass that had some of the characteristics of wheat and made it more nutritious, more palatable, easier to what's it called when you beat the wheat? Thrash. Easier to thrash, getting the kernels out of it. You want to learn more about that? You can look in your book, or you can take my Bio 2 class. It's actually kind of an interesting concept of genetics, especially among plants. Okay, so alterations can occur to a structure of a chromosome, right? I'm sorry, to a number of a chromosome. You can either have one too many, one too few, or maybe a whole set, or you can have alterations to a structure. Now, when is this going to happen? It can happen during synapsis. So synapses can, instead of crossed over in the right way, they cross over backwards. It can cross over something we're going to talk about in subsequent chapters when DNA makes copies of itself, during mitosis or meiosis. Cosmic rays. Thank goodness for our atmosphere, and our not our atmosphere, but our magnetic atmosphere, our magnetic shield that keeps a lot of the cosmic rays from space from getting down here. But they're high energy, like little pellets, man, and they come at near the speed of light and they bounce off your DNA if they just happen to hit you by really random chance, and they can screw up the molecule. They can mess it up. Now, many times when you mess up a molecule, these little things, these little enzymes, proteins, they go along the molecule and they try to fix it. But sometimes they don't fix it perfectly. It's like, like patching a hole in the wall. If you don't do it right, you always kind of see where the patch was, or they fix it wrong. And sometimes what they do is they remove part of it, or some part of it gets removed, so you have a deletion. Now that's a problem. It's not a problem if this happens in the part of your chromosome that isn't red. Remember, I called it junk DNA for lack of a better word. It's not all really junk, but the part that doesn't get turned into proteins. Or essentially what we call blind mutations or silent mutations. Silent mutations would be all of this on a page, these deletions or anything that occur to a part of your chromosome that you don't use. Remember I told you that most of years ago we found out that only about 5 of your chromosomes actually get turned into a protein. Now your chromosomes and your genes have lots of other functions that we'll talk about in other chapters. Like, for example, there are genes that regulate other genes and inhibit other genes. So they don't make proteins, but they do control genetics. But if you got a deletion in one of those junk parts, you say, it wouldn't be an issue. But if you deleted the lactose gene, right? If you deleted the gene that helps you make the enzyme lactase, so actually it's more than one, but the enzymes that digest milk sugar, you'd be lactose intolerant. There's probably many reasons why folks are lactose intolerant, but that may be one of them. B is the Goldilocks syndrome, having too much of something. Actually, if we go back for a second, in trisomy 21, the phenotype that you see, the manifestation of the disease, the learning disabilities, is because on the 21 gene, there's a GART gene. I don't know if that's an acronym for something, but they, like I said, they name genes. And if you have trisomy 21, you have 3 GART genes, right? You don't have 2, like all the rest of us have 2. I think that's partially the problem with trisomy 21. The GART gene makes too much of something. There's 3 of them. Unfortunately, if something happens during a replication or some mutation occurs and you get an inversion, then part of the genetic information is in there backwards. And unfortunately, your cell reads your genes the kind of the way you read a book, right? It reads it left to right. So it's like if part of your book was cut out and the letters were reversed and stuck back in there. Now, does that end up making a protein structure? Well, you'll learn in subsequent chapters that maybe it does or maybe it doesn't. But if it does, it's backwards. And if the primary level of protein structure is so important, you got something messed up here, don't you? Okay, so it can be gone, it can be duplicated, it can be backwards, or it could be not gone, but in the wrong place. In a translocation, what happens is something gets screwed up during synapsis. All right, so during the time when homologous chromosomes are supposed to come together, like let's say this is the number 8 paternal, something gets messed up in synapsis. Instead of synapsing with the number 8 maternal, it synapses with the number 3 maternal, a totally different chromosome. For some reason, those magnetic genetic parts allow them to stick together. Some mess up, obviously, so they undergo synapsis, they cross over, and when they do, they happen to swap, in this case, these genes. So this is called a translocation, because what you've done is you've removed a gene from one area, one chromosome, and you stuck it on another. And in this case, you stuck it at the end, all right, but what if you stuck it in the middle of another gene? It'd be like reading your book on genetics, right? You're talking about aneuploidies and polyploidies, and then all of a sudden we're talking about the battle for Rome or something. And you're like, well, what? And then you jump back into, translocations. You're like, where did that come from? It's like maybe you have friends do that. They talk about one thing, and then boom, all of a sudden they talk about another thing, and you're like, what? I had a friend of mine in the military years ago. I finally got to the point and said, you have to say new topic, because I'd sometimes lose track of what you're talking about. Now, unfortunately, in our cells, the cells don't know, the cell machinery that's going to make the protein doesn't know it's been messed up. I mean, you might be able to go, oh, come on, this has nothing to do with genetics, the Roman history. But in your cell, when it gets ready to read the gene, your cell has kind of like an index book. Make me gene X. Oh, okay, where's gene X? The machinery says, well, it's on the number 8 paternal at the tip. Read the genes there. Oh, gosh. So it'll read them, and maybe it makes a protein, but whatever, sometimes it doesn't, as we'll learn, but whatever protein, if it does make, it's going to be misshaped. Then, of course, on the other chromosome, the same thing happens. So I get, if you think about it, of all of these, which one would, I don't know, it depends. I guess a deletion might be, well, any of these that occur during the junk genetics, it's not going to mess up anything. But I guess if you have something that's not, I don't know. Translocations probably are particularly damaging. You can see translocations too. It's really cool. Remember I was telling you about making things glow? It's such a neat part of, in microscopes, it's all about staining things so you can see them, right? Remember when we, in the microscope lab, when we stained the onion with iodine? Remember how much clearer that made the picture? So this whole thing of making things glow or fluoresce, oh man, I tell you, you get awesome pictures. Here's a picture of a translocation. So these are chromosomes all in a karyotype, and we're taking a picture of them. Now, you're going to get some random colors because of genetic information. So ignore this red over here, but look at these two here and here. Same colors, same chromosome, same genetic information, same sequence of letters. So the ones that are red are homologous, the ones that are blue-green, they're homologous. But look at the tip of the blue-green one and look at the tip of the red one. They have a color of each other on each other, right? There's a little bit of red on the blue one, a little bit of the greenish blue, well, I guess it's mostly green on the red one, isn't there? So these are live chromosomes, if you will. Isn't that cool? Neat pictures. I love that stuff. Okay, folks, I think I owe you a few minutes anyway, because I know I've ran over a couple of times. So that's going to be lecture for today.